We now turn from the non-invasive techniques employed in research on living human brains to more direct techniques, which are commonly employed in biopsychological studies of laboratory animals. Stereotaxic surgery, which is a procedure through which experimental devices are positioned in depths of the brain, requires the use of a stereotaxic atlas, which is like a map that you'd see up here. This would indicate how far the specific location is. Additionally, we would then hold the mouse in place and make sure that the drilling is with the precision that's provided by the atlas here. The stereotaxic instrument, which is here, has two parts: the head holder, which firmly holds each subject's brain in the prescribed position and orientation, and an electrode holder, which holds the device to be inserted. A system of precision gears allow the electrode holder to be moved in three dimensions: the anterior, posterior, dorsal, ventral, and lateral, medial. Eventually, the electrode is anchored onto the skull with several stainless steel screws and dental acrylic that's designed to harden around the electrode connector. Once the electrode's in place, we are able to see the effect of certain stimuli on the brain and also measure electrical activity in the brain. In these methods that we'll be looking at, a part of the brain is removed, damaged, or destroyed. Then the behavior of the subject is assessed in an effort to determine the functions of the lesion structure. We'll start off by looking at the aspiration lesion. If you speak Spanish, you'll know that aspiration actually has to do with suctioning of, and like a vacuum, aspiradora. And this is basically this is used with easily accessible cortical areas to the surgeon's eyes and instruments that are drawn off by suction. Please note that this is done on the cortical tissue, and because it's the underlying white matter that is slightly more resistant to suction than the cortical itself, a skilled surgeon can, for the most part, delicately peel off the layers of the cortical tissue from the surface of the brain without damaging the white matter and major blood vessels underneath. With radiofrequency lesions, small subcortical lesions, sub meaning below the cortex, are commonly made by passing radiofrequency current, and that is a high frequency current, through the target tissue from the tip of a stereotaxically positioned electrode, just like we showed you earlier with the rat. The heat from the electrode will destroy the tissue, and the size of the shape of the lesion are determined by the duration and intensity of the current and the configuration of the electrode tip. The third one, which is a knife cut, is perhaps the most straightforward, and I'll show you a picture in an upcoming slide, and that is where there is a cutting that is used to eliminate the conduction of a nerve or a tract. With a tiny, well-placed cut, we can unambiguously accomplish this task of cutting without producing extensive damage to the surrounding tissue. In the next slide, we'll show you how we can insert a knife into the brain and make a cut without severely damaging the rest. Please note that lesion effects are deceptively difficult to interpret because the structures of the brain are small and very convoluted and packed in a way that's really difficult to separate them, that even a highly skilled surgeon cannot completely destroy a structure without producing significant damage to adjacent structures. As a general principle, the behavioral effects of these lesions are difficult to interpret. For the most part, unilateral lesions, which are lesions that are restricted to one half of the brain, are much milder than those of symmetrical bilateral lesions, which are lesions involving both sides of the brain, particularly in non-human species. They find the behavioral effects of unilateral lesions to some brain structures can be difficult to detect, and as a result, most experimental studies of lesion effects are studies of bilateral rather than unilateral lesions. Last, we will look at the cryogenic blockade, and this is the only one of the lesion methods that's reversible. The way this works is we temporarily eliminate neural activity by freezing it. With body temperature increasing thereafter, we would defrost this specific part of the brain and it would gain its functionality. Please note that when I initially recorded this, I did not have a picture up. And just as promised, here is the picture, but it would appear for you since the beginning. Here is the knife cutting picture through which a surgeon is able to release a blade underneath and it would cut the tissue of the brain as noted here. Electrical stimulation is another form of invasively studying physiological responses in our brain. This lesion can be used to remove damage or inactivate a structure, and it is done while the patient is awake and aware. Because the brain has no pain receptors, it is uncomfortable. The patient does not feel the insertion of the electrode, and this is often done to treat Parkinson's disease and other neurological disorders. The electrical brain stimulation is usually delivered across the two tips of bipolar electrode. These are two insulated wires that are wound tightly together and cut at the end. The weak pulses of a current produce an immediate increase of the firing of neurons near the tip of an electrode. The electrical stimulation of the brain is an important biopsychological research tool because it often has behavioral effects, usually opposite to those produced by a lesion to the same site. It can also elicit a number of behavioral sequences, including eating, drinking, attacking, copulating, and sleeping. The particular behavioral response elicited depends on the location of the electrode tip and the parameters of the current and the test environment in which the stimulation is administered. Because electrical stimulation of the brain is an invasive procedure, it's limited to non-humans. However, there are situations in which it is administered to conscious human patients. For example, with Parkinson's patients who have severe tremors, and this has been done to stop these tremors. In order for us to detect any changes or effects of this, we have to be able to record the electrophysiological activity. And so we'll look at 4 invasive methods as to how this is done. First one, as the name implies, intracellular unit recording, is with a microelectrode that's positioned inside a cell, specifically a neuron, and this would provide a moment-by-moment record of the graded fluctuations in a neuron's membrane potential. Most experiments using this recording procedure are performed on chemically immobilized animals because it is difficult to keep the tip of a microelectrode positioned inside a neuron of a freely moving animal. Extracellular unit recording is where a microelectrode is positioned outside of a neuron, and it does the same but from the outer component. This records the electrical disturbance of a firing neuron. With multiple unit recording methods, we have a small electrode that provides a record of firing of multiple neurons. And so it uses a larger electrode tip, which then picks up signals from many neurons and the disturbance that a firing neuron causes. The last of the 4 is the invasive EEG recording, and this is where a large implanted electrode rather than a scalp electrode. And this picks up cortical and subcortical measures. This is done in laboratory animals, where EEG signals are recorded through these large implants